All right. Hi, everyone. My name is Kelsey Zielinski, and this is my second year working as a graduate mentor in the P2EP program. I am a PhD student in NC State, working under the direction of Dr. Marianne Lila and Dr. Massimo Urizo, both of whom bring complementary strengths to my thesis, which has the goal of bridging the gap between the nutrition and genetics field so that not only can we identify compounds that are beneficial for human health from plants, but we can also breed for those compounds in the crops of interest. So we have 3 projects going on this summer, and the overarching goal, as I said, is to develop resources to characterize the anthocyanin pathway in blueberry. But first, I'd like to introduce my interns. This is Maddie McLaughlin of Brigham Young University, Jacob Goodnight of NC State, Justin Stevenson of NC State, Michaela Graham of NC State, and Jacob Ferrier of UNC Charlotte. So I know you learned a little bit about Blueberry this morning from Samantha's group, but Blueberry has such a high regard on this campus that we felt it deserved 2 projects. So we're working together towards a common goal. And blueberries are packed with secondary metabolites known as anthocyanins, and these anthocyanins are thought to act as antioxidants within our bodies, which means that they actively scavenge free radicals or neutralize free radicals, And this is important because high concentrations of free radicals in the body can lead to states of chronic inflammation, and this condition is associated with other diseases such as heart disease or arthritis, certain types of cancers. Some of the research projects we've had going on this summer, let's take it back a step. So even though blueberries have such important impacts for human health, there are very few genetic and genomic resources that exist in order to study blueberry. So the long-term purpose of my project is to develop genetic resources that will allow us and future researchers to better understand or elucidate the genetic mechanisms behind these traits with an emphasis on anthocyanin biosynthesis. So we've been working on 3 projects this summer. The first one is involved with developing a method that is suitable for characterizing anthocyanin content in blueberries and suitable specifically for genetic studies. So the current method is based on a per gram whole berry weight. And the issue here is that the potential problem here is that anthocyanins are expressed exclusively in the skin of blueberries, and it's actually what gives them the blue color of blueberries. So if you were to compare 2 cultivars, one with small berries and one with large berries, you might find that the cultivar with smaller blueberries has a higher anthocyanin content, but this may actually just be due to the fact that they have a higher surface area to volume ratio as opposed to actually being a genetic mechanism. So we first want to identify if a bias exists for this method, and then if so, how can we remove it? The second component of this project is to make one that is high throughput, because genetic studies often employ screening an entire biparental mapping population, and these populations are commonly several hundred individual bushes. So if, for example, you were to start off with a population of 365 individuals, and they're analyzed in triplicate at 2 sites over the course of 3 years, this very quickly amounts to over 6,500 samples. So as you can imagine, we really need a highly throughput way of screening large numbers of samples. So once we've identified a method that can be used for quantifying anthocyanins, this can actually be employed in Project 2, where we're seeking to identify an actual genetic resource in the form of a biparental mapping population, specifically one that is segregating for anthocyanin content. So if we can identify a population such as this, then we can use it to map regions of the genome that contribute to the anthocyanin biosynthesis by screening all the individuals within the population. But the story doesn't end there, because once you've identified regions of the genome that contribute to that trait, you need to further determine what gene or genes within that region of the genome contribute to the anthocyanin biosynthesis. So this leads to Project 3, which is sort of a parallel effort to Project 2, where we're searching through the literature in order to identify papers that have already validated the function of genes within the anthocyanin pathway in other species, and then we can download these sequences and use them to predict genes of the same function in the blueberry genome. So once we identify these regions, then we can combine this or compare this data to the data that we produced in Project 2, and we hypothesize that those genes that we've identified from Project 3 will overlap with the regions that we found from Project 2, and those regions are otherwise known as QTLs, in case you hear that term later. So by combining these projects in parallel, we can expedite the process of identifying the actual gene or genes that contribute to the anthocyanin biosynthesis. And with this knowledge, we could actually employ it or be used in a blueberry breeding program to increase the anthocyanin content of said blueberries. So with that, I'm going to turn it over to my interns, and Maddie's going to tell you more about Project 1.
Thank you, Kelsey. Like she said, I'm involved with Project 1, which is a methods-based approach to investigate anthocyanin content in blueberry for genetic studies. So the first question that this project asks is there a bias associated with the traditional wholeberry method of anthocyanin quantification? To solve this, we harvested 2 bushes and separated the berries by size into groups, large, medium, and small. And because we're interested in the relation of anthocyanin content to surface area, we used an image technology to extract the surface area called GINA. It was developed by a group in Wisconsin, and we were able to take the surface area from that and then with the extractions retrieve the anthocyanin content, and that's what you see in the graph on the bottom of the slide. And as you can see, as you go from large berries to small, both the surface area and anthocyanin content increase. And so that indicates that there is a bias associated with this method. And because that bias exists, we're seeking to eliminate it. So we're developing what we call the squeezeberry method, and that involves separating the skin from the pulp and then extracting anthocyanins only from the skin. And by doing that, the data we retrieve should be suitable for genetic studies. So I'm going to let Jake explain the second objective.
Thank you, Maddie. Our second objective was increasing throughput in our methods. As Kelsey mentioned, it's very important to increase throughput in genetic studies because you have to analyze a large number of samples. Thankfully, having developed the squeezeberry method, where we isolate the portion of the blueberry that contains the anthocyanins, the skin, we have concentrated the anthocyanins in our sample to the point where we can analyze them with near-infrared spectrometry, NIR. However, before we can use NIR, we had to develop what's called a calibration curve to translate the raw spectral data into usable data. So we receive raw spectral data from 57 genetically diverse genotypes that we have analyzed, and it's titled Original above me. This raw spectral data needs to be converted in pre-processed into the pre-processed form shown above, which reflects merely the chemical components of the sample. This pre-processed data is then correlated to traditional HPLC measurements of anthocyanins to create the final calibration curve shown on the right. Our calibration curve has an R-squared value of about 74%, which indicates NIR is a viable method for anthocyanin quantification in blueberries. Now I'm going to turn it over to Justin, who's going to talk about our second project.
All right. Thank you, Jake. Our second project is entitled The Characterization of a Genetic Resource for Mapping Anthocyanin Accumulation in Blueberry. So in addition to these anthocyanin quantification methods, our project also requires a genetic resource from which we can extract information as to map regions of the blueberry genome responsible and that contribute to anthocyanin accumulation in blueberry. So the genetic resource we settled on is known as a biparental mapping population, and that entails selecting 2 parents that differ for a particular trait of interest. In our case, one parent that has a high total anthocyanin concentration and another with a low total anthocyanin concentration. These parents are then crossed, and you follow up with the progeny and analyze them for that particular trait of interest, in this case, total anthocyanin concentration. Now, as Kelsey mentioned, a biparental mapping population typically involves several hundred individuals, and in order to circumvent the necessary time and resources that would take, we instead selected a subset of 50 progeny from a pre-existing biparental mapping population and analyzed those for total anthocyanin concentration. Now, the results of those efforts are seen on the bar graph on the screen, with the total anthocyanin concentrations of the parents in orange and that of the progeny in green. As you can see, there's an approximate 3- to 4-fold difference in the total anthocyanin concentrations of the parents, as well as that of the lowest total anthocyanin concentration of the progeny to that of the highest concentration. Now, this, combined with the low overall standard deviation in these values, indicate to us that this population is likely segregating for total anthocyanin concentration, and as such, we feel confident moving forward with this population as our genetic resource in our future efforts. And I'm now going to turn the microphone over to Michaela so she can inform you about the other objectives in this project.
So for our second objective for this project, we wanted to know that since the HPLC data shows that this population is likely segregating for total anthocyanin content, will the NIR high throughput method also show the same results? So in the graph above me, you can see along the x-axis is the NIR values, while the y-axis shows the HPLC values. And the line through the center has an R-squared value of 0.91, which indicates that these two data sets are highly correlated. This tells us that we can use both HPLC and NIR to screen the rest of the population for total anthocyanin content. The last observation in this project is that now that we know that anthocyanins are likely segregating in this population, are there other metabolites that are also segregating within the population? So we sent off 100 genotypes to be analyzed from global metabolite analysis using NMR data, or NMR methods. Now, the other Blueberry group this morning also mentioned that they used NMR data in their study, and just to clarify, this is NMR data from a different population. So out of this NMR data, we found we identified 7 metabolites that also seem to be segregating within this population. And these are shown above me in the 3-dimensional graph. This graph shows all the 7 metabolites that we found, along with the concentration values for the samples, the range of them on the front x-axis, and then the number of genotypes or individuals along the y-axis that had this range of concentrations. Within these 7 metabolites, we are interested in chlorogenic acid because of its similarity of health benefits to anthocyanins, such as antioxidant, anti-inflammatory, and anti-diabetic activities. In the future, we may do a mapping assessment for these 7 metabolites. And now I'm going to turn it over to Jacob, who is going to talk to you about our third project.
Thank you very much, Michaela. Our third project, an integrative genomics approach to predict genes in the anthocyanin biosynthesis pathway, has 2 major parts. Our first part, as Kelsey mentioned before, is us attempting to find as many functionally characterized genes within the flavonoid biosynthesis pathway, which you can see in the center screen above, as possible. Thus far, we have found 143 functionally characterized genes within this pathway. However, our primary mode of focus is within the green part of the pathway, the bottom, the anthocyanin pathway, and particularly one enzyme within the anthocyanin pathway, the acyltransferase enzyme. Which leads us to question 2. Could sequences of validated anthocyanin and acyltransferase genes be used to identify orthologous genes in the blueberry genome? To accomplish this task, we found as many acyltransferase enzymes as we possibly can within the bottom of the pathway. Acyltransferase, as the name suggests, is an enzyme that transfers an acyl group onto an anthocyanin, which effectively acylates the anthocyanin. Literature suggests that acylated anthocyanins are less bioavailable in the human body as non-acylated anthocyanins, meaning you would theoretically require more acylated anthocyanins to get the same nutritional benefit as a non-acylated anthocyanin. This sounds like we would obviously want to breed out for acylated anthocyanins. However, before we do this, we need to establish some genomic resources and actually establish where it is on the blueberry chromosome, or blueberry genome within a chromosome, and that was the latter part of our project. So to do this, we downloaded protein sequences of 17 acyltransferase genes that we found within the flavonoid biosynthesis pathway. From there, we did an orthologous analysis between these 17 downloaded protein sequences and the recently constructed blueberry genome by the Rizzo lab. From there, we found 2 distinct clusters, one for the BAHD family of acyltransferase and one for the SCPL acyltransferase family. Both these families accomplish the same task, acylating anthocyanins. However, they pull from different substrates to get their acyl groups. So you can see as figure A, right above me, the 12 blueberry chromosome schematic. And you can see highlighted chromosome 8 and chromosome 9. Chromosome 8 is where we found the BAHD cluster of enzymes. And within chromosome 8, we found 5 different genes representing 20 isoforms for acyltransferase genes. And on chromosome 9, we found 2 genes for the SCPO family representing 39 different isoforms. So all this data suggests that we were able to answer a preliminary question. Yes, we are able to pull sequences from other known genomes to functionally validate acyltransferase genes within the blueberry genome. This leads us to Kelsey, who's going to tell us a bit more about future plans.
Thank you, Jacob. So some of the future work for these projects, the first one is that we'd like to increase the number of samples used in our NIR calibration curve so we can have more of a powerful prediction for a wider range of samples and increase that R-squared value. For the second project, we would like to evaluate the total anthocyanin content for the remainder of the population since we only screened a subset, and now that we know that it is segregating, we're going to continue screening the remainder. And then again, we can use this phenotypic data along with genotypic information from the population in order to locate the QTLs or regions of the genome that contribute to this trait. And for the third project, we need to continue collecting more genes or functionally validated genes from within the flavonoid and thus anthocyanin pathway. And then, as I mentioned, these can be used for the orthologous analysis to determine whether those regions overlap with QTLs identified from Project 2. And with that, I think we encourage you to come visit our posters and ask us questions if you have any. Thank you.